Sorry.
All right, here, if you're videoing me, if you want to just like prop it up here
No,
or something.
that's okay. I just wanted to leave.
Okay, that's going to just see the ceiling the whole time anyway.
Yeah,
okay. All right, but anyhow, though, so the plants phylogeny lab, if you have not yet finished that one, make sure I get that from you next Monday whenever I return. The pre-lab on the next plant activity on flowers, fruits, and seeds, that will be the lab activity that we do the week I get back. If you can complete that pre-lab before then, that would be good. And of course, the thing for this week, you've got two things to do while I am out of town. One of them, of course, is your visit to Phipps Conservatory. I didn't have a chance to run off physical copies of the assignment, but the assignment is available on Blackboard. Really, you don't even need to print out the actual physical assignment. You can do it on any piece of paper that you want, but you might want to check it out and make sure you know what is expected of you for that. Let me reiterate, though, go to Phipps, explore around. There are a bunch of different rooms at Phipps. Visit at least 7 of the different distinct rooms there. I've listed a bunch of them on the assignment, but if you want to go to other ones, that's cool too. Go to at least 7 distinct rooms. Find at least 10 plants that you like. Give me both the common and scientific names of the plant in question. Give me a description of the plant. Tell me about, the features it has that, make it distinctive and identifiable. And then tell me about an adaptive feature of the plant. Tell me about something that that plant has going for it that helps it to adapt to the conditions of its environment. Again, I'm interested here in ecological, not economic features. I'm not interested in what we as humans do with the plant. I'm interested in what features that plant has that makes it better able to cope with the world in which it dwells. Also, when you're noting the common names of the plants, a thing that you should pay attention to is that a lot of the plants in the conservatory, and this is a tradition in conservatories that goes back to like the 1700s, that a lot of times they name their plants. Like a plant that's been in the conservatory will have a name. So it's not unusual, to find a plant that it will say like, common fern, Furnus longiva, and underneath it will say Pappy, or like whatever other name they've given that particular fern. I don't need to know that the fern is called Pappy. I need to know common fern and Furnus longiva. That's what I need to know. So when I say give me the common name and the scientific name, I'm not talking about whatever nickname they gave the plant. I don't care if it's called Carl. That's not. I don't need to know what its friends call it. Yeah, so pick any 10 plants you like from at least 7 different rooms, so obviously some rooms you'll take multiple plants from. Pick interesting plants. Pick plants that you got something to say about, and again, to find out about adaptive features, some of that may be just a matter of looking at the plant and applying a little bit of common sense. Some of that may be a matter of asking questions of the people at the conservatory about the plants or looking on the conservatory's website for data about them. Or even doing a little bit of side research on them later. It might be a matter of reading the information they have about them. The conservatory is not like the Carnegie Museum in that not every plant has a little placard detailing, what its features are and what it's all about. But there are some bits of literature available around the different rooms. Pay attention to those. They can tell you a lot of useful things. And take your time. Honestly, Phipps Conservatory is a really awesome facility if you've not been there. It's one of the nicer conservatories in North America. And it's well worth spending a little bit of quality time in to visit and explore and see all the cool things they have on offer. And if you like it, it's one of those places, go back other times of the year. They change the exhibit seasonally with different kinds of plants being in bloom. So it'll be a different experience in summer or fall than it will be right now. All right, so do you guys feel like you have a handle on that? Yes?
Which pre-lab are we doing?
I would like you to finish the pre-lab on flowers, fruits, and seeds. And then finally, the other thing I would like you to do whilst I am out of town is if you go on Blackboard in the assignments folder, you will find an assignment on microevolution. And I would like you all to complete that. It is mostly a matching assignment, where I've given you examples of different ways that organisms have adapted or evolved, and you tell me which evolutionary phenomenon they match. Like, is this an example of, negative frequency dependent selection? Is this an example of heterozygotes advantage, whatever. Yes?
You said that was due next Monday?
That's all these things I've just described to you are due whenever we get back. If you can't make it to Phipps for whatever reason on the actual lab day, get it to me as soon as you are able. But keep in mind, this is your lab activity for while I'm gone, so try to make it there sometime this next week. All right, sir?
Will the completed fungus lab be next Thursday? Or
actually, that was the next thing I wanted to go to here. First off, let me ask you all a question about the status of your fungi right now. How are they looking? Does anybody have full coverage on their Petri dishes yet? Almost? You're getting close? Most of you should be getting close to full coverage. the fungi are not typically going to grow a lot more beyond, like, say, about 2 weeks, and we're about close to hitting the 2-week span. So what I would like you all to do is to continue to take observations of them the next day or two. I will be here tomorrow. I'll unlock the lab in the morning, or if you find it unlocked, just drop me a message, and I will come and unlock it for you. I will have a word with the secretary up in the department. I'm told that some of you went up today and tried to get her to unlock the lab when you found it locked, and she was just like, I can't do anything about it. I will have a word with her and let her know that in that case she's supposed to call security and tell them to come and unlock the lab for you, So even when I'm not here, if the lab is locked, I will ask somebody to open it for me in the morning, and when they fail to open it, you can just call security or through the secretary and have them come and open it up for you so you can get in there and do your observations. But keep on observing your fungi through Wednesday. And then I feel at that point we'll have a good 2 weeks of observational data. We can cut it off at that point, and then on Monday I will give you all a sense of what I'm going to expect from that lab report. I have not put together the full assignment yet. Maybe I'll even post it to Blackboard between now and then if I get the opportunity. But frankly, I'm getting ready for a conference. I have a lot of shit to do right now. I may not actually get that done before then. Everybody have a sense of what all is likely to be happening while I'm gone? Got an idea what I expect of you and so forth? Okay, cool. Okay, so today is our last class for this week then. I will not be here Wednesday or Friday. I have not been able to get anybody to sub for me those days. I will, however, post a digital lecture on something. We'll see how far we get today, and then I'll have a better sense of what that's likely to be about. So watch that before I get back in lieu of class for Wednesday or Friday as well. But I'll make an announcement on Blackboard when I do that. So just be watching your email for those announcements too. All right, but let's pick up where we left off, see how far we can get with the time that is available to us. Last time we gathered, we were taking a look at the phenomenon of macroevolution. We talked about some reproductive barriers that can cause isolation in populations. We began describing a couple of different ways that speciation can go down. We have now mentioned allopatric and sympatric speciation. We examined how allopatry is largely a consequence of geographic isolation. We started talking about ways that sympatric speciation can happen, in which somehow you can get isolation even within organisms that live in the same physical territory. And we outlined an important way that can happen the other day in the form of non-random mating. As a mechanism for that. We also talked about polyploidy as a way that can happen. So before we move on, did anybody have any questions about anything from last week? Sorry.
You had actually completed the lecture without discussing non-random mating?
Oh, we've gotten it that far, but we haven't talked about it? Okay, we'll get to that first. Thank you for letting me know.
You get the red and blue back cichlids.
Okay.
You get the red and blue back cichlids.
Yeah, right. Yeah, that's pretty much where we're at there. But yeah, any other questions about stuff from last week? Okay, one last thing about the whole polyploidy thing. I told you guys the other day that polyploidy is normally a thing that happens with plants much more than it does with animals. I actually have an article that I'll be putting on Blackboard for you all to check out in the near future about a really cool developments. You can probably go on NPR's website and find the article that is happening right now in the Galapagos Islands. Specifically, we have witnessed an allopolyploid appearance of an entirely new species of Galapagos finch within a 2-generation span. You all have heard about the finches in the Galapagos Islands by now. One of the Galapagos Islands is an island called Daphne Major. Daphne Major has been the site of ongoing evolutionary research that's been happening there for more than 30 years. A couple called the Grants have dedicated their lives largely to observing evolutionary phenomena on Daphne Major. They visit the island a couple times a year. They survey the finch populations that live there. Well, what happened is not too long ago, some migratory finches moving from one place to another within the island chain ended up on Daphne Major by mistake. That's there was like a storm or something blew them off course, and finches that don't normally travel to Daphne Major ended up there. What happened is that some of those finches interbred with a local finch on Daphne Major, and what we ended up with was a new hybrid species of finch that is not cross-fertile with either of its parent species, but is cross-fertile with other finches of its kind. So there's now this terribly inbred little population of completely new hybrid finches on Daphne that represent a wholly novel kind of Galapagos finch. So it's a rare example of allopolyploidy in animal populations. and one that has appeared literally while we watched, you know. So it's a really exciting event, and to have it happening, you know, in the Galapagos seems especially poignant and all. So I will put the article on about that on Blackboard. You know, it's a pretty nifty situation.
Are we going to Daphne Major for the trip?
We will not be going to Daphne Major for the trip. Daphne Major is uninhabited. It's extremely isolated from the other islands. And it's actually really, really fucking difficult to get onto Daphne Major because it's this very, very tall rocky island. Like when you get to the base of it, you actually have to climb like 40 feet up to get to the actual surface of the island. There's no beach on Daphne Major, so accessing it is very, very hard. You need a lot of special permissions from the government of Ecuador to get there, and frankly, you need some good rock climbing training to get there. So we will not be visiting Daphne Major. We will be sailing past Daphne Major, and I will point it out to you guys as we go past it on the boat. But we will not be visiting that one, no. I would love to, but it's really just not feasible. All right. So carrying on, though, we've learned now that sympatry is the thing that we have to address. How exactly we can get reproductive isolation within the same physical zone that will nonetheless create the conditions needed for novel speciation. We've seen that polyploidy is an easy way to achieve that simply by rather rapidly cutting off organisms from gene flow from others that are no longer compatible with them. But there are a couple of other ways that we can make this happen. One of them, of course, is non-random mating. Now, you all know, because we have discussed sexual selection, that mating is seldom completely random in any case. There are usually going to be mate preferences that are going to happen, and there are going to be cases in which organisms differentially pick one kind of mate over another. That often leads to the appearance over time of sexually dimorphic traits, you know, like bless you, the females prefer males with the big fancy tail feathers in the peacock, for example, stuff like that. Now, if that's directional selection, if that is a case in which the preferences are always favoring a particular kind of traits, that tends to lead to variation within the same species going in a particular direction. Microevolutionary change where it's like, okay, we always favor the male with the biggest feathers or whatever. Now, however, let us just suppose we end up with a mutation which causes some of the females to differentially prefer a different kind of male. That rather than having the entire population trending after one particular variant, where it's like, okay, we always like the biggest crests, you know, or we always like the brightest feathers or whatever, let us suppose that we have a bifurcation, that we have a case of disruptive selection in which some of the females really prefer males with one kind of trait, and some of them really prefer males with another kind of traits. If that happens, what you can end up having are two parallel breeding populations existing side by side in the same territory, where the females of one are going to strongly prefer the males of a particular kind, and another group of females will prefer a different kind of male. As a result, you can end up with genetic isolation, reproductive isolation, simply because they don't recognize one another as mates. This is all a case of what we would think of as behavioral isolation, where the female is differentially selecting males for a certain desirable quality. That might be something like ornamentation. You know, again, the classic example, the blueback versus the redback cichlid. The female prefers the male with a certain scale color. This might be a behavioral thing. I gave you guys, I believe, the example of the Congolese river eel, you know, in which some of the females are not able to detect the frequencies at which some of the males are communicating, and now we have two parallel eel populations based on which ones can pick up on one another's electrical signals. Whatever the instance, whatever the reason is, once we have two separate groups that are selecting for different things, we now have a case in which the population has basically split in two. And they're now two separate gene pools even within the same zone. Now, presumably, a lot of the same selective pressures are still bearing upon them. You know, if eel 1 and eel 2 are living in the same river, pollutants in that river are going to affect both of them. But without genetic exchange between the two groups, the solutions arrived at to those problems may be different. And mutations that help one of those groups may not be the same mutations that help the other one. And this can lead to a situation in which part of the population splits off and begins doing things a new way, or a situation in which both parts of the population begin adapting in novel ways to selective pressure. And this can mean leading to either the appearance of a new species while the old one continues to plug along, or it can lead to two new species arising while the original basically is phased out. Right, does that make sense? Now, lastly but not leastly, if leastly is a word, in terms of how we can achieve isolation sympatrically, another way that we can make this happen I think this is the most interesting way of having sympatry take place, is through what we call habitat differentiation. Habitat differentiation is a situation in which the split in populations happens because some portion of the population begins exploiting a new niche in the environment. That again, that this is a form of sympatry that happens when a portion of the population begins exploiting a new niche in the environment. Yes?
Sorry, just for continuity. Last time you had numbered autoploidy, alloploidy. Would this be number 3?
This would be number 3. Yeah, basically we've got polyploidy, we've got non-random mating, and this would be habitat differentiation, a third way we can get sympatry happening. Thank you for clarifying that. This usually happens when the environment changes in some fashion, and most commonly when a novel resource is introduced that was not previously there, when something new comes into the environment, and a portion of the population begins adapting around that new thing. The organisms that begin adapting to the novel resource will typically begin accumulating variation that makes them better suited to that. This can lead to a variety of types of reproductive isolation. My favorite example, New England, 1700s. The British colonies are thriving there, and they are bringing stuff from the old world. You know, they're not just relying on the native resources, they are bringing along crops and animals and so forth and things that they know from back home. One of the things that was brought to the Americas in the 1700s, first introduced in New England, was the apple. Apples are not native to the Americas. They were brought here by British colonists. Now, in New England, if you go to New England even today, you will find lots of hawthorn trees. The hawthorn tree is one of the big native trees up there. Hawthorn trees do produce a fruit. It's not a very good fruit, but it's a thing that they produce. You know, the hawthorn fruit is the thing that's available up there. And there's a native insect called the hawthorn fly. The hawthorn fly lays its eggs inside of hawthorn fruits. And then the maggots hatch within the fruits. They basically have an immediate food supply available to them as soon as they hatch. You know, they spend the first, couple week or two of their existence, munching their way through the medium that they hatched into, accumulating nutrients until they mature into a fully formed fly. Now, once the apple tree is introduced, an interesting thing happens. Some of the hawthorn flies begin laying their eggs in apples instead. This is a novel resource that some of them begin to exploit. What's interesting here, and why this is important, is that hawthorn fruit and apples mature at different times of year. As a result, what happens is that little by little, the breeding season of the flies that are exploiting apples begins to shift around the apple blooming season. And therefore, you end up with two populations of flies in New England, the hawthorn fly and the so-called apple maggot fly. And the apple maggot fly, this is a charming name for you, the apple maggot fly has a breeding cycle that is largely based around when apples are mature and available. That is not the same breeding cycle as hawthorn fruits. So as a result of this, we have temporal isolation. The animals are no longer breeding at the same times of year. Their mating seasons are out of sync now, and as a result, we have two parallel breeding populations. One population of flies that continue to feed on the hawthorn fruit, another population of flies that are differentially feeding on apples. And as a result, now we have these two different species exploiting different niches within the same physical territory. Yeah?
But wouldn't there have to be an overlap between the maturation cycles of hawthorn and apple trees?
There's not a lot of overlap there, but what ended up happening basically is that some flies that otherwise would have been out of the breeding pool altogether because their natural mating instinct was for a time of year when hawthorn fruit were not available, basically had something to work with. Essentially this required a mutation in the, like any evolutionary event, this required a mutation that was going to be useful in a given environmental circumstance. In the absence of apples, the fly that had the mutation that shifted its breeding cycle more towards apple season would have been a very unfortunate fly. That would have been a fly that, unless it found something to lay its eggs in, would have been out of the gene pool altogether. But in the presence of abundant apples as fruit that they could lay their eggs in, and an instinct that told them lay your eggs in fruit, the fly that had a mutation that caused its breeding cycle to shift to later in the year is one that now had a thing that it could do. All right, does this make some kind of sense? So you can see through situations like this, by exploiting different habitats within the same physical zone, exploiting different resources in that way, or by having differential mate choice, it is possible for us to get parallel breeding populations within the same territory. And once that reproductive isolation has been established, whether it's behavioral, whether it's temporal, whether it's something else, those populations begin to accumulate their own genetic variations. And in the fullness of time, that is going to lead to them, or can lead to them, splintering off and forming novel species. Does this make sense? Now let's talk about how fast this happens. How quickly does speciation occur? One of the favorite arguments that is regularly dusted off by anti-evolutionists is, well, this is all speculative, we've never watched this happen. Aside from the fact that that is a flat-out lie, because we've watched speciation happen many, 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 many times, including very recent examples like the one I just told you about that we literally watched happen last year in the Galapagos. We've witnessed speciation happen in real time on